Good afternoon, and thank you very much for the invitation to speak to you today about BovReg and how we hope the results will impact our production in Canada, our beef production in Canada. So you can see that we're part of this European project. This is the group of the team that is working on the project at the University of Alberta in Livestock Gentech, and I want to show you what beef production is like in Canada, or in Alberta a little bit at least, and show you why the results of the project will be so important. So just a reminder, BovReg is looking to improve the annotation of the bovine genome, of course, and the key traits we're looking at in the project are biological efficiency, health and robustness, and you'll see that those are the traits that we're certainly very interested in beef production in Canada. So again, our hypothesis, if you like, is well identified in the latest FAANG paper from Emily Clark and colleagues, From FAANG to Fork, and I've just highlighted this statement. Chief among the improvements required is the ability to more accurately use genotype to predict phenotype in specific environments. And that is illustrated by the work from Australia, from Iona McLeod and colleagues, who you all know very well, in this paper that was published in PNAS. So by focusing on causative mutations, we will overcome some of the barriers that we see to adoption which are related to the accuracy of prediction. So just to get our bearings, here are some numbers from Canada, and you can see the average size of beef operation is 147 head per farm, and that varies across the country. Alberta, where our group is based, is a little bit more, and you can see that around 41% of the beef cows in Canada are in Alberta. And you can see that at the beginning of this year we had approximately 3.5 million beef cows and about 1 million dairy cows, but I'm focusing on beef today. And one of the things I should say is that whilst we often think of genetic improvement in terms of breeds and breed associations, that of course is part of what we do in Canada, but the commercial cow herd represents crossbred animals. So 95%, approximately 95% of these cows are crossbred, and of course that's one of the challenges for us in terms of applying prediction to improving the output from our commercial cow herd. So just to come back to G2P in different environments, then here's some examples of how we feed cattle in the winter, and we graze them. So these cows are generally outside 365 days of the year, and at least 150 of those days in our part of the world are winter grazing. So they have to be well adapted to the environment that they find themselves in. And of course in the summer and spring and summer the grazing varies as well across or up and down the province, depending on the type of forage that's available and how we feed. So what we're doing is driven by the National Beef Strategy, and you can see this is the latest version of that, and you can see some of the targets we have: improving the top cutout yield, AAA if you like, the best eating quality animals, a 2% reduction in open rate, so 2% increase in calf crop percentage, so around reproduction improvement. Despite all of the improvements that we've made in nutrition and reproductive technology, actually our calf crop percentage is stagnant at the moment, and it's lower than we would expect it to be. So this is an important challenge for us. And of course, as for everyone else, when we talk about ruminants and cows, we'd like to improve feed efficiency and reduce the impact of livestock on the environment, including the reduction of greenhouse gas emissions, as well as reducing mortality. So that is identified as these are the things we want to achieve, and one of the ways that it talks about doing that is capturing phenotypic and genomic data along with additional information for commercial cattle, as I've talked about, and thereby enhancing the accuracy and value of genetic evaluations for both the seed stock sector and that big sector of commercial producers, the 3.5 million cows. So how are we going about that? Well as FAANG and BovReg, we've been building our reference population for crossbred beef cattle, and this is an effort that involves both the University of Alberta, Livestock Gentech, but also Agriculture and Agri-Food Canada, and some of the people you saw in the first slide work for Agriculture and Agri-Food Canada at the University of Alberta. So we have genome sequence on more than 350 bulls, which are part of the 1,000 Bulls project. We have nearly 5,000 HD genotypes which we've imputed to genome sequence, and if you look at that, we have 27,000 genotypes which are imputed up to 43 million SNPs. In terms of phenotype, we have more than 12,000 records of dry matter intake, average daily gain, residual feed intake, more than 8,000 carcass traits, and nearly 3,000 with shear force records. And then on the fertility side, more than 10,000 mating opportunities to look at fertility and genotype. And in terms of greenhouse gas emissions, we have been using green feed to collect more than 30,000 spot measurements for enteric methane and CO2. So increasing all of the time and building up that reference population to be able to do G2P. So I just wanted to briefly talk about the animals which are part of BovReg. So our part of the project is providing animals for FAANG assays from the Kinsella crossbred, and you can see this is the average breed composition of our herd. The current herd is around 400 cows, and you can see the biggest component is Angus, perhaps not surprisingly, because we try to follow commercial trends. Next is Charolais, then maybe surprisingly Galloway, and then Hereford, and you can maybe see your favorite breed here or work it out. And so we use our genotypes to predict breed composition, and here you can see some analysis for some purebreds, and not surprisingly they form separate clusters. And then this group in the middle here includes our Kinsella crossbred. You can maybe see some of the orange dots, and then this is another. The bigger cluster is a series of composite maternal lines which are used in Alberta. So you can see, as you would expect, our breed is laying on top of that distribution. So those are the animals we're using in the BovReg project. But I just wanted to come back to how we're using genomics for commercial cattle. So we're using that database to predict 18 molecular breeding values for 18 traits, and you can see our accuracy is ranging from 0.35 to 0.45 depending on which of the traits we're talking about, and progeny equivalents of 10 to 20 calves per bull. So quite a good data set. And what we've been doing is looking at validating those predictions, and we've done that with 2,000 crossbred feeder cattle with carcass traits from 152 sires, and we've also tested some of those through GrowSafe in terms of their feed efficiency and growth. And Cargill have provided, or we've collected from Cargill, those carcass traits, including the price paid for them and the premiums and discounts. So a very nice data set, and I just want to share those results. And you can see here's an example of, we call it feeder profit index, and it's the MBV, so the DNA based tool. You can see the different weights that we put on these traits in the index we're using 6 carcass merit traits, post weaning average daily gain, of course very important to us here. And this is one example, a couple of sires here with an index of minus 163 compared to one with 208, and you can see the average profit per feeder from these bulls, and every 100 unit change in the FPI resulted in an increased profit of $29 per feeder. So it's working, let's say that. And what I want to do here is pause, but just remind you, adoption of these technologies is of course dependent upon the accuracy and also the cost. So the BovReg project, we hope will help us continue to improve the accuracy of our predictions, and then we'll also be working on different ways of reducing the cost and implementing this technology across Canada, but and of course in Alberta. And these are the members of the team contributing to the molecular part of BovReg, making sure we've got the right phenotypes and the right tissues for our FAANG assays. I'll stop there. Thank you very much.